our next presenter is Stefan, and he's going to be talking about MetaNeighbor. And feel free to start whenever you like.
Thanks, Joe. Sorry, just let me start my timer. So I'm sorry, I'm recovering from my cold, so my voice is a bit broken. So I'll try my best to keep up with the talk here. So thanks a lot for giving me this chance to present MetaNeighbor in front of this audience. I'd like to say before I start that compared to the previous tools, MetaNeighbor is very focused on a single task. So we've seen all these tools with so many different tasks you can handle, and then those nice browsers to display the different results they get. With MetaNeighbor, we're really diving into one problem for which we wanted to find a simple solution. So it was about finding a simple way of quantifying cell type replicability, compare different cell types across data sets with a tool that I hope I'd show you is very simple to use. So I'll spend a lot of time demoing the tool, but I'll give you a couple of slides to introduce what MetaNeighbor is, how it works, and how we used it in the BICCN. So before talking about what we did in the BICCN, what does MetaNeighbor do, and what do we have in mind when we developed that tool? So the key question we were interested in is we have two data sets that define two seemingly completely different sets of cell types. So imagine if the authors are different, they won't necessarily have the same names for their cell types. And so we wanted a way to say, okay, this cell type A in data set 1 matches with cluster 17 in data set 2. And this is a surprisingly difficult problem. So let me just give you an example. If you have a collection of data sets with cell types, so I picked here an example with 7 data sets, and I was careful to pick 6 cell types that are very different from a biological point of view. So it should be very easy to match them across the 7 data sets. The first thing you could think of is, I just take the correlation profiles. Like if they're use the same gene expression programs, then if I correlate the transcriptomic profiles of the different cells within the cell types, they should match up together. I should see high correlation values. So this is what was done here. And you could see it's quite disappointing. And the reason for that is that correlations are very quickly eroded by what are called batch effects. So there's a lot of technical variation in the cell types that interferes with the gene expression profiles. And so this will have strong consequences for these correlation computations. So what we were aiming to do when we developed MetaNeighbor is, could we have something that looks like correlation, but goes across this issue, so removes the technical variation and only focuses on the biological part. A second constraint we had is that we didn't want to integrate the data set. We wanted the tool to be simple to use, so simple from the user's perspective, but also computationally, so really not have to work on the data set, on the pre-processing, and just have something that works. And so this is the output we get with MetaNeighbor on this data set. I hope I can convince you that we get really clean, perfectly correlated the clusters once we switch to the MetaNeighbor similarity. So I'll give some mathematical details in the next slide. I just say a few words about why we're interested in developing MetaNeighbor. There are 2 key applications. One is comparisons to a reference data set. So having worked in the BICCN, we have a lot of collaborators that come to us that have neuronal data set and say, okay, I've clustered it. I don't know what the cell types are. Can you match it to the BICCN reference data sets and just call what the different cell types are? So with MetaNeighbor, we have a very quick way of matching the cell types of our collaborators with BICCN cell types. So they have standard names for each of the cell types they found. And the second one is about identifying replicable types. So this is more about finding consensus types. So this is more what was done within the BICCN. Is if we have lists of cell types that don't match across a lot of different data sets, can we subset it to types that are found independently in all the data sets and for which we know there will be a strong biological basis for replication? So a couple of words on how MetaNeighbor works. So how does it get around the batch effect? So it uses an algorithm that's called neighbor voting. The way it works is that we always consider only a pair of data sets. So I gave you an example with 7 data sets, but truly we'll only consider 2 data sets at one given iteration of the algorithm. And more specifically, we'll pick one reference cluster in the first data set we're considering. And then what we're going to ask we're going to use a prediction framework and ask, is there a cluster in the dataset 2 that corresponds to the reference cluster I just picked? And the way this works is we have the cells in this reference cluster vote according to transcriptional similarities. So they will give strong votes to the cells that they're closest to in terms of gene expression, and only low votes to cells that are quite far away. So each cell receives a group of votes. And what happens is that if there is a cell type that overlaps well with the reference cell type, it will have all the strongest votes. And if it's far away, only have all the weakest votes. So if we ask, if we have a strong overlap between two cell types, what it means is that from a prediction point of view, if I only focus on the strongest votes, the reference state cluster will be very easily predict that this cluster D2 is the matching cluster in dataset 2. Whereas all these that are further away will have weaker votes, and so they won't be predicted as being the closest cluster. So it's an algorithm that's quite simple, that's very efficient to compute, which means that it is almost as fast as computing correlation in practice. So you have to apply this for every pairs of data sets and every pairs of cell types you're considering. But in the end, you can group it all together in a matrix and similarity matrix that shows you which cell types correspond across the different data sets. So you just have one matrix in the end that tells you A1 matches with D2, B1 with B2, C1 with C2, and it recognizes that A2 has no match in dataset 1. So MetaNeighbor was used in several studies in the PICCN. So here I'm highlighting the single cell omics working group that Iran presented at the beginning of this session, where the working group generated 9 different data sets using almost as many different single cell omic technologies. And so we were interested in one specific iteration of the analysis of these data sets. So that's when people were analyzing those different data sets independently. And we were wondering, so what's the set of cell types that we find even before we integrate all those data sets together? So how many cell types do we find by clustering those data sets independently? And so by running MetaNeighbor, we found that there were 60 neuronal clusters that had very strong evidence of being found in the best powered data sets. So in multiple best powered data sets. I don't show it here, but there are other applications where we use the same approach to find cell types that are conserved across different species. So in another study where we compared marmoset, a mouse, and human, we use MetaNeighbor to find sets of cell types that are conserved throughout evolution. And we also use MetaNeighbor to find the gene sets that are conserved. So there's the basis, the biological basis of why these cell types are conserved. So I'm going to switch to a demo to show you like how MetaNeighbor is run in practice and how you could use it on your own data sets if you're interested in these types of approaches. To make it focus on the ACCN data, I decided to pick 3 out of the 9 data sets from this single cell omics working group. To make it simple, I only pick smaller subsets of the sample. So I only focus on GABAergic cells, and if there are more than 5,000 cells, I randomly don't sample those to 5,000. And I picked 3 technologies that are quite different. So not necessarily easy to match: the single cell SmartSeq, single nuclei, 10x version 2, and single cell 10x version 3. So if you'd like to follow along or reproduce what I'm showing here, so I'll show you all the code that was used to generate the figures, so you have an idea of how easy it is to use MetaNeighbor. So it's really only a couple of steps. But if you'd like to follow along, or at sometime later reproduce the analyses, maybe use it on your own data sets, tweak it a bit, the full notebook can be found on GitHub, so at the link shown below. It's in R Markdown, so if you have a decently recent RStudio installation, you can just download the notebook, and everything that's needed for the demo will be downloaded automatically within this notebook. So the first step is just installing MetaNeighbor. I just wanted to point out that it is based on Bioconductor. So if you're used to using Bioconductor, it's a very simple one-line install. If you don't like Bioconductor for some reason, you could just install it from GitHub. So all the data I've downsampled for this demo I've put in specific files that you can get from Figshare, so those will be downloaded automatically. I just wanted to point out again that this is not the official data that was used in the working group. It's a smaller version of the data. So the labels are the true labels that were found in the group, but it's really just a small version. So if you want to use it for a real study, consider using the full data set. The first thing to know about MetaNeighbor is because it's part of Bioconductor, it uses single cell experiment as its input format. So you need to have a data set, a single cell data set that's formatted in this type of object. If you're used, for example, to Seurat, it's very simple to generate a single cell experiment object from Seurat. So it's just a simple function call. The point of having a single cell experiment object is that you have one object that stores the count matrix, but also all the metadata that's associated with this count matrix. So in this case, I've removed a lot of the metadata. I've just kept labels and sublabels. So these are two different levels of the hierarchies of neurons defined by the BICCN. But you could also store something like UMAP coordinates or t-SNE coordinates. So the first constraint when you run MetaNeighbor is that you need to have all the data sets in a single cell experiment object. So the first step is using MetaNeighbor to merge the different data sets that you consider in your approach. So you just have to provide them as a list. And the point of doing this is that it will make sure that all the metadata and the gene information aligns across the different data sets. So this function will automatically pick the genes that are common to all the different data sets. Oof, I'm trying to get my mouse here. So there's 24,000 genes that are common to those SmartSeq and 10x data sets. In total, we have 12,000 cells, and we have 3 metadata columns. So they're the labels and the subtype labels that were inherited from the single objects. And this function will also create a new column that will tell you from which data set the cell originated from. So all these cells, for example, originated from the single cell SmartSeq data set. And so once it's done, so the data is formatted, there is no normalization needed at all when you run MetaNeighbor. So it doesn't matter. You don't need to do anything like CPM or scaling. We switch to computing highly variable genes and then computing the MetaNeighbor similarity, which is quantified as an AURC. So the highly variable gene selection, we have a function in MetaNeighbor that does it for you. You just have to provide the data, the slot in which the count matrix is stored, and then the labels for the data set. So the way this function works is it computes the highly variable genes independently in each data set and then finds consensus highly variable gene. But feel free to use any highly variable gene function that you like from any other package if you prefer. And then you just run MetaNeighbor. Again, very, very simple parameters, which is just the set of highly variable gene, the merged data, where the count matrix is stored, the labels of the different studies that were used, and then the cell type labels that you want to compare. And there's a final argument that is about using a fast version. So MetaNeighbor was developed quite a long time ago in 2018, before integration and stuff like that were even invented. And so it was run on very small data sets. So the legacy version was not optimized. So we kept it for compatibility reasons. But if you want something that runs fast, so in order of seconds for 10 to 100,000 cells, you should use the fast version. And then you get this output. So you can most easily plot it as a heat map. So MetaNeighbor will take all the cell types in the different data sets that you've fed it and give you this similarity matrix. Usually what you're looking for are red blocks, like clear red block structure, which we get here. And usually the red blocks should be the size of the number of studies you're considering. So for example, this is a 3x3 red block. This is exactly what we want. And if we look at what the cell types are, we have one cell type that comes from each of the different study we considered. And we have here the author-defined labels, which are SNCG, SNCG. And the last one, for fun, I just decided to remove the cell type labels and hide them and see if we could match back what the original labels are. And here I bet really hard that CT5 was originally SNCG. So this is expected. Those cell types are very highly replicable. So this is something I knew before. And you can see how this works perfectly, like VIP, LEM5, SST. So you get all the big classes of GABAergic cells. And visually, it's very salient that they replicate perfectly across all of the data sets. But what's nice with MetaNeighbor is that you not only match the cell types across data sets, you also get a value of how strong the matching was. So AURC of 1 is the best score you can get. Essentially, it means it's a correlation of 1. And everything down to ARC of 0.9, we consider to be excellent replicability. So this is the case for this data set. Just to give you an example of something that goes wrong, it's very important to keep in mind this ARC threshold and not just match anything with anything. So this is the results of the function call I've just shown you. And then after the second one, we've permuted the labels in one of the data sets. So here it means that for this SN10X data set, there shouldn't be any matches because I just permuted the labels. And indeed, they really only weakly match to the different cell types. But just be careful. Don't think that CT2 and CT4 may be related to PV and SST because they're on the same part of the tree. It's quite important to also check that the replicability value are high. They could just be associated to the cell types randomly. So I'll switch to a slightly different metric. And with time, it will become clear why we define this. So this is something we developed while we're working on the BICCN data. And this is something we call 1 versus 1 or 1 versus best AORC. So the pipeline is pretty much the same as before. You compute highly variable genes, you compute the meta-neighbor similarities, and there is a new option here that says I want 1 versus best AORCs. So this was the previous result. This is what we call 1 versus all AORCs. And what's nice with those is that you really get the comparison between each pair of cell type in the data set, meaning that you only get local matching, you also get global structure. For example, if I look at this NCG, I see how it nicely replicates across all the 3 studies, but I also see how it's related to the VIP type. And then it's also not so far away from the CLAMP5 type. And taken together, those are all the CGE-derived cell types. And same thing on the other side, PV tends to be related to SST, and those are the MGE-derived cell types. So we also capture those global relationships. However, if you're really interested in one-to-one replicability, like does my cell type match very precisely to the other cell type in the data set, then we switch to 1 versus 1 AORC. So here we lose global matching. So we only asking, can I separate that cell type from its closest related cell type? So we lose a lot of the global information because we're not even considering these comparisons, but we refine the replicability values so they become much more stringent. And I'll show you in the next slides how this VIP versus SNCG replicability drops because the boundaries of those cell types is not exactly replicated across all the data sets. So a very small shift in boundaries here is caught up by this 1 versus 1 versus best AORC. One nice way to represent this matrix, because it's now mostly empty, is just to plot it as a graph. And this gives you a very quick idea of what's going on in your different data sets. So the one structure you want to look out for are cliques. So these are groups of 3 cell types coming from each of the 3 data sets with the corresponding colors. So each color here is a different data set. And you have these groups of perfectly replicating types. So the gray arrow means that this cell type believes that this red cell type is its best match. And so these are reciprocal hits, and there is no secondary hit that they consider to be even remotely as strong. So this is true for SST, LAMP5, and PV, but it's not true for VIP and SNCG. Here, it seems to be that the boundary is not exactly right. And if you look at the cell types involved, there's this SN10X data set that seems to be involved in all the miscalls. So it seems like the boundary problem might be between those 2 subclasses. So a bit more about that in a couple of slides. And I haven't really told you why we developed this in the first place. So we wanted something that was more stringent, but the real reason is that once we consider 9 different cell types, each containing hundreds, 9 different data sets, each containing hundreds of cell types, it just becomes impossible to just look at these types of matrices. Like the 1 versus all will still give you the global information. You'll see the MGE CG split, but at this point, we're not interested in MGE versus CG. We just want to know, do those fine grained cell types replicate across the different data sets? So with the 1 versus 1, we get a more precise picture of what's going on locally. And then we start seeing some structure like here, this 3x3 red block that indicates that this cell type replicates very well. And again, the matrix here is really hard to follow because there's so much information and so much empty space. So we switched to this graph representation where we really have a better idea of what's going on. So you, again, can really look for the cliques And what's interesting is that if you start looking at the names, I mean, they match perfectly across all the different data sets, and there is a reason for that, because those cell types are cell types that were previously known. Like we've talked about these a bit in this session, like SST shuttle, which are long projecting interneurons, PVL Viper 2, which are chandelier cells, for examples. One clique that's interesting is this one. You'll notice it has two SNCG subtypes that have slightly different names in the SCSS and SS single cell 10x data set, but they're strongly replicating in this single nuclei 10x data set. But in this data set, it was labeled as a VIP subtype. So this subtype, just because it's a real cell type, but there was slightly inconsistent annotation at this stage of the analysis, such that it was labeled as a VIP type, and this messed up a little bit with the VIP versus SNCG replicability. What's also interesting to look at are cases where things don't replicate as well anymore. If you look at LAMP5, it looks like it's always groups of 3 different colors that go together. So this is mostly indicating of good replication structure. The boundaries again are not perfect, but that's okay. And then we have more extreme cases like the VIP subtypes, where what's striking is that in a single nuclei data set, there were only 2 subtypes here that were found for a lot of the different single cell 10x or SmartSeq types. So you kind of get a picture of what's replicating well, and when it's not exactly replicating, is it more like an end-to-end matching situation, or is it more like I have real cell types but the boundaries are not exactly defined? You can also try to get rid of this mess and really focus on what's working. And so we have a function that extracts what we call meta clusters, where we really focus on the one-to-one matches. So they will give you the top hits, the cliques that were found in this graph structure. For example, the SST shuttle is by far the best cell type that was found in all studies, and always with a replicability score of 1. So you get a score for all of these potential cell types that are replicating. Then there's a lot of cell types that only replicate in 2 studies, which is expected because the single nuclei data set was not has high quality, and then a list of cell types that had no correspondence in the other data sets. So I switched for the last 5 minutes to a slightly different version of MetaNeighbor. So this is a tweak where we see it really as a second step. The first step is about matching cell types across data sets, find replicable cell types. And the second part is what's driving this replicability? Why are the cell types replicable from a feature point of view? What gene sets are driving this replicability? So here there's a strong constraint that the cell types need to match across data sets before you run the supervised MetaNeighbor. And this is why we call it that way. So there's a first step where I made sure that all the names are exactly the same in all 3 data sets. If this is complicated because the names are quite different, you could use the meta clusters I just showed before as a way of having the same names across the different data sets. Then we need a list of functional gene sets. So here we're removing the highly variable gene selection that was the first step before, and taking genes that have biological significance. So here I'm just taking all gene ontology sets that are between size 20 and 50. 20 because I want the signal to be robust, so I need a minimum set of genes to compute the correlations between the cells, and 50 as a maximum because I want the function to be quite specific. The function call is almost the same. You know, this is just called MetaNeighbor. Before it was called MetaNeighbor US because of MetaNeighbor and supervised. And we're feeding the gene ontology sets instead of the highly variable genes. So this takes much longer to run. So in this case, before you could really export the data and run MetaNeighbor a couple of times on different gene sets, different versions of your data. It's all done in seconds. Here, because we have to go through 2,500 gene sets and basically run MetaNeighbor each time, you should expect this to run for minutes or even hours for large data sets. So in this plot, we're showing the overall distribution of contribution to replicability. So each tick is a gene set. And the higher it is on this scale, the more it contributes to making this cell type replicable. So like this gene set alone would be sufficient, for example, to define the SNCG subclass. So on average, we get something that's very close to 0.6, 0.65 performance, which is very low contribution to cell type definition. Here, we consider that the threshold of 0.8 is a good indicator of a gene set that contributes a lot to cell type identity. An easy way to look at this is you can look at the different values across the cell types and then look at the average. And so this helps you find gene sets that contribute a lot to of the identities of all the different cell types you're considering in your data set. And here you see that glutamate receptor activity is the top performing gene set before a lot of gene sets that are either representing axon guidance or channel activity, which are all expected functions in the context of neurons. We have a function that helps you understand why these gene sets were at the top from a gene level point of view. So for example, here, this gene set that was axon extension involved in axon guidance, you can see how there are a combination of genes that drive replicability. So for example, those SEMA genes are semaphorins, and you can see how each of those neuronal subclasses uses its own grammar of semaphorins, essentially. So they have their own combination of semaphorins. So you could explore the key gene sets using this function. So the summary is, I hope I showed you that MetaNeighbor is a very scalable tool and that you can really easily compare data sets at a cell type level using that. The key thing is that we don't use integration, which makes this as fast as correlation, but we remove the batch effect that you could typically see if you use correlation directly. You can use it either to compare a data set to reference data sets or extract replicable cell types. And once you have those, you can find the gene sets that contribute to replicability using the supervised version of MetaNeighbor. I hope I've convinced you it's not browser-based, but it's still simple to use. You need to have RStudio, of course, but then it's really just, 3 steps if you count merging the data sets, but then it's really just highly variable with gene set selection and then computing the RSC, and then a couple of visualization functions. If you're interested in MetaNeighbor, we have a publication that shows how MetaNeighbor works and a couple of examples, but we also have extended protocols that look a little bit like what I've shown today. So really hands-on examples with BACCN and non-BACCN data that show the different functionalities of MetaNeighbor and that were published last year in Nature Protocols. So feel free to email me with any questions, and thank you for your time.